Hi class, Dr. Jim here. In this lecture we're going to be looking at cell division, what it's called, mitosis, and another process which is called cytokinesis. And cytokinesis means the equal division of cytoplasm between the two cells. And we're going to be looking at both processes today. Both are really important, obviously, when you want to divide your cells and go from the process of one cell to two. And we're going to be looking at that 10% of that stage that I talked about in the last lecture, where we had interphase and then mitosis. Interphase was at 90%, basically getting the cell ready to divide. And today we're going to focus in on the 10% of when the cell divides. All right, and so here's the lecture. And so first of all, I'm going to show you what the chromosome looks like. And the reason why I do this is because the chromosome looks really different at interphase than what it does when you undergo mitosis. And there's a reason for that, because you want your DNA to divide evenly, and so you want to make sure you get the right pieces going to the right thing. And so it really will make sense once we start going through the phases why it needs to condense into what it looks like. We're then going to look at how the cells divide evenly, and so we're going to talk about mitosis, what takes place, how does the cell get ready, all these different things, what does it need to do in order for these things to happen. We're then going to spend some time looking at the stages of mitosis, making sure that you understand what's going on in all these different stages. I think to be ready for the next exam, make sure you know all 4 stages of mitosis, what takes place at all 4 stages, and then also being able to identify using a microscope slide, identifying what stage that cell is undergoing or what stage of mitosis that cell is at when you look at it under the slide. If you can do all those things, you're going to be really ready for the next test. And then finally, we're going to talk about cytokinesis. And the reason why we're bringing this up as a second point or a third point in this lecture is because cytokinesis is different between animal and plant cells. In animal cells, it divides with the cleavage furrow, which is kind of this pinching of the cell and then pulling apart. Whereas in plant cells, they don't have that room to pinch and pull. And so what happens is they actually form a cell plate in the middle and then divide their cells evenly by forming a wall in between the 2 new daughter cells. And so we'll look at that more specifically here in a few minutes. All right? So that's where we're at. So again, the chromosome structure, the DNA is a very long piece of a long thread. And I think I've used this example before, but if we took a cheek cell from you and we popped the nucleus and pulled out one of your chromosomes and stretched it from end to end, it'd be about 6 feet long. So in order for all that DNA to be compacted into that nucleus, we need to have mechanisms in which we can fold and coil and put those things in. And the way we do this, as these chromosomes are called, is we wrap these things in histone proteins. And this histone protein wraps the DNA up into these tight little packages so we can put the DNA into these nice little nuclei and packages so that they can be stored in the cells. Now, what we call DNA and histone proteins collectively, we call this chromatin. And chromatin looks different when the cell is in interphase than what it is during mitosis. Now, chromatin looks like a big ball of DNA during interphase. And what I mean by that is basically if you took the cover off of a baseball or maybe a golf ball or some other, or maybe like a rubber band ball, and you see all those strings wrapped around in different directions, that's basically what your DNA looks like in the nucleus. It's all kind of wrapped together, no real organization going on in that case. Okay. However, during the process of mitosis, that DNA actually condenses into what we know as chromosomes, and you guys have all seen this before. They look like these X-like structures. And the reason for this is that we want to make sure that our DNA divides properly. And if it's left as a big tangled ball, you've always dealt with Christmas lights or whatever, and a tangled ball, very difficult to do. But if we can condense it down into these little structures that we can pull apart, makes it so much easier to deal with, and that's why we do that. Okay, so chromatin is a big ball of DNA during interphase and then condenses down into these chromosomes so that they can easily divide during mitosis. All right, and so we can look at the numbers of chromosomes per organism, looking at their counting how many chromosomes they have during mitosis, and we can see that humans have 46. Okay. Now, I never want you to associate size of an organism or intelligence or anything like that with the number of chromosomes, because there's a wide variety of different types of chromosomes, how many organisms have, and it varies from species to species. And there really is no rhyme or reason why some have more than others. It's just how the DNA is aligned, how much intron, exons they have, and things like this. But here's to that point. Yeast, which are really small, you have to look at it under the microscope, only have 32 chromosomes. I mean, they almost have as many chromosomes as we do. Whereas if you look at something like a fern, a plant, this has 1,320 chromosomes. Or let's take our average ordinary goldfish that we have swimming in our tanks. That goldfish has 94 chromosomes. It actually has twice as many chromosomes as we do, but it's much smaller and it's not nearly as intelligent as we are. Okay, at least we can make that claim. And so if you look at these things, you can't use the criteria of intelligence or size or any of these other things to see how many chromosome numbers, because that really isn't the key. What the key is that each species has their own set of chromosome numbers. Okay? Now, there are some definitions that I'd like you to know, and we're going to be using these definitions not only in this chapter, but in many other chapters to follow. And this is really important, so I would really write this down and make sure that you understand what these two definitions mean. The first one is diploid, and diploid means the total chromosome number. So in humans, that number is 46. If you remember back to the slide previously, we talked about the goldfish and having 94. Its diploid number would be 94. That fern that had 1,320, that's the diploid number. Now, the other definition is haploid. Haploid means half the number of chromosomes, or the number of pairs that you have. So in humans, the number of pairs we have is 23, or half the number of chromosomes we have is 23. And why does that make sense, or why does that work, is because we get 23 chromosomes from our mom and 23 chromosomes from our dad. And we put those together, we end up with 46 total. If we took the goldfish and looked at its haploid number, that haploid number would be 47. So 47 come from the goldfish's dad, and 47 come from the goldfish's mom. They come together for baby goldfish and make 94. And that's how we look at these things. So diploid means total number of chromosomes in the cell. Haploid means half the number or how many pairs you have. Okay, so make sure you keep those definitions straight. All right, so now we're getting our way into mitosis. We looked at the previous lecture and looking at the different phases. And again, interphase is about 90% of the cell cycle, and that includes the G1, S, and G2. And again, it does not proceed into mitosis unless all these things take place. If you've grown your cell big enough in those two growth phases, if you've duplicated your DNA properly, and that your cells are ready to divide. Your DNA is condensing and things like this. If you get the go-ahead signal, you then can proceed into mitosis. And again, mitosis has what I like to call 4 phases. Now, in your book, they like to say 5 because they kind of say this pro-metaphase is a separate phase. But I don't like to overcomplicate things, and all I want you to know is the 4. Prophase, metaphase, anaphase, and telophase. And in a minute, I'm going to show you the different phases and what goes on in those different phases. But for right now, what I want you to know is, again, 90% interphase, the rest of the time's mitosis, and again, there's the 4 main phases of mitosis, okay? Cell has to get ready in order to do that. All right, so what happens to the chromosomes? So I've talked a little bit about the structure of the chromosomes and how they condense during mitosis, and that it makes them easier to pull apart. Well, this is another thing that I would definitely write down in order to make sure you keep this straight, because this can get easily confused by what is what and what is something called at what time. And so let's take a look at this. So at the end of the S phase, when you duplicate your DNA, each chromosome is internally duplicated. And what does that mean? Well, basically you have 2 strands. This strand is the old and new strand of one DNA, and this is the old and new strand of the other DNA. And so you open those up. You remember from replication, you open up the strands and you make one new strand, and so you have the semi-conservative. That's what this chromosome represents, okay? So this is called a duplicated chromosome, this structure here, duplicated chromosome, and each one of these is called a sister or chromatid, okay? Sister chromatid or one chromatid, however you want to describe it. And during mitosis, what happens is that these chromatids will get split apart. And as soon as they split apart, the one old new get split apart from each other, they now become individual chromosomes. Okay? So this is an important spot right here. Each becomes an individual chromosome. So during mitosis, you're going to see this split. And as soon as they split apart from one another, they no longer are chromatids, but now become chromosomes. Okay, so that's the hard thing to remember because this whole structure is a chromosome, duplicated chromosome, with individual chromatids, but as soon as they split during mitosis, these now become individual chromosomes. So keep that straight. All right? All right. So just outside the nucleus, we have these structures called centrosomes. And what centrosomes have are basically these centrioles. And I talked a little bit about centrioles before when we looked at eukaryotic cells, and I said basically their function is to help divide the cell. And that's really all I want you to know. What they do is they provide anchors on either side of the cell to help split it apart. So think of like a tug of war where you have the two big guys at the end and they anchor the tug of war so that you have that. And that's basically what's going on. These centrioles act as the anchors to help pull the cells apart. And that's what takes place during mitosis. So that's what the centrioles do. What I want you to know about centrioles is that they act as anchors, help divide the cells into two, and that during mitosis what happens is that you'll see these centrioles migrate to either end and help pull the cell apart because they start at the nucleus and then work their way out to the poles. And we'll talk more about that when we get into mitosis. Okay? So let's look at the phases of mitosis. The first phase of mitosis, once the cell is ready to go, so we say, okay, cell, you're ready to divide now. You've made the criteria of growing big enough, you duplicated your DNA, and you're ready to go. What happens? So the first phase is called prophase. And again, what takes place is that chromatin, that big ball of DNA, condenses into the individual chromosomes. And you can see that here. So what happens, that ball of DNA becomes the X shape that we see. And so this ball of DNA in here becomes these X chromosomes. Now in this cell representation, it shows 4 chromosomes. Now I want you to kind of remember that in your mind as we go through this. 4 duplicated chromosomes, okay? So we have 4 duplicated chromosomes. In humans, we'd have 46, okay? Just to kind of give you an approximation there. The next thing that happens is you see this nuclear envelope, it disintegrates. We lose that nuclear membrane because we need to get the DNA outside of into the cytoplasm in order to divide the cell. So we can't have the inhibitory effect of the nuclear membrane around. So what happens is that disassociates during the time of mitosis. The two centrosomes move away from each other, so the centrosomes are by the nucleus, and what they do is they move towards the poles. Now, I also want you to think about the cell being a globe, okay? And you have the 2 poles on either end of the cell. So you have the 2 poles, and then you have an equator in the middle, okay? And so we talk about the globe or the globe effect of the cell. What happens is one centriole will move to one end, and the other centriole will move to the other end, and the cell can then begin to divide. What we also see are these spindles, and the spindles are really the fibers that help pull the cell apart. So the anchors are the centrioles, and these fibers that attach to the chromosomes will help pull the cell apart. And I'll show you here in just a minute. So prophase is really the cell getting ready for this division. You lose the nuclear membrane, your chromatin condenses into chromosomes, getting ready for division, and your centrioles move to the poles. Those are the 3 main things that I want you to know that go on here. Okay? The next stage is called metaphase, and metaphase really is the representation of this. If you think back to your gym class days when you had your gym teacher with a whistle, he'd blow the whistle and tell you guys to line up. Well, that's really what the cell says. It says, okay, cell, chromosomes, line up, and what they do is they line up on this metaphase plate, and you can see this here. The 4 chromosomes, the duplicated chromosomes, line up on the equator, and these centrioles, which are on either pole, will then begin to pull these guys apart. And you can always tell metaphase in any cell, either animal or plant, by looking for the chromosomes all lining up in the center. If you see that line up in the center, you know that's metaphase. So always think line in the middle, metaphase. Okay? That tells you metaphase on that equator. The next stage is called anaphase. And anaphase is when the chromosomes that, you remember when you had the duplicated chromosomes and you had the individual chromatids, they begin to be pulled apart. And as soon as they get pulled apart, they're now individual chromosomes. So you remember that? I told you chromosomes, the whole thing is a chromosome. When you pull apart, now they become chromosome 1 and chromosome 1. Okay, they're individual chromosomes. Okay, the sister chromatids separate, and they're now called individual chromosomes, and they're now pulled to the opposite ends of the cell. And if you see this space between chromosomes like this, you know your cell is in anaphase, and that is the dead giveaway of anaphase. You start to see this pulling away, this pulling part of the chromosomes, and the space in between the chromosomes, that is a dead giveaway for anaphase. So always remember that when you're looking at your slides and looking at those things. Now the next phase is called telophase or telophase. You hear it both ways. In telophase, what you see is that the cell is now beginning to look more like what the cell did at prophase. And so what we see is that the spindles begin to disappear. You now have two clusters of daughter chromosomes. Okay, now what's important, remember when I said at the start at prophase, remember that number? That number was 4, right? You had 4 duplicated chromosomes. So let's count how many chromosomes we have in the cell right now. We have 1, 2, 3, and 4. And in this cell, we're going to have 1, 2, 3, and 4. And so remember, the diploid number is the total number of chromosomes you have in a cell. We started with 4 duplicated chromosomes, and the diploid number was 4. And we maintain that number. Now we have 4 individual chromosomes per cell. We're going to have 2 cells. So that number is maintained. So remember that. Mitosis, that number, diploid number is maintained. We don't change numbers of chromosomes or anything else. You start with 4, you end with 4 in each cell. Okay? The other thing that happens is you start to see the nuclear membrane reform, starts to form back, and the cell starts to look like the cell again. The other thing that you start to see in these cells is you start to see the cells dividing. And this is called cytokinesis. And I'm going to talk more about this in just a second. But what that is basically where you get this division or invagination of the cell membrane, and it starts to pull and spread these cells apart. In plants, it's a little bit different. What you get is what is called a cell plate, and that starts to form in between the two cells and forms a wall between. It's like building a brick wall between the two neighbors to separate the neighbors out. And we'll talk about that in just a second. So cytokinesis is equal to the division of the cytoplasm. Okay, so dividing the cytoplasm means cytokinesis. Okay, what this does is it means that the mother cell divides its cytoplasm equally between its two daughters. Okay, and in animal cells, this forms a cleavage furrow, this invagination, where you dig in and pull. You invaginate the cell and pull like taffy. Think of it that way. It's like pulling on taffy or silly putty. When you pinch in and pull, that's what happens to the cell. And eventually it breaks and it snaps the two into 2 new cells. Now this is different in plant cells because plants don't have the room because they got these really structured cell walls around them. And so they can't pull apart. So what they do is instead of pulling apart and invaginating from the outside, they start to build a wall from the center. And so what you see is you see the cell plate forming in the middle, and eventually it becomes one solid wall, and eventually it ends up to the end. So think of inside to out. So cell wall builds from inside to out, whereas in animal cells that pinching begins from outside to in. And so that's the difference between the animal and plant cells. So make sure you understand the difference. Plants form the cell wall in between and they work from inside out, whereas the animal cells pinch from the inside and pull apart. And that's how we divide our cells. Now here are all the phases, and you remember I told you at the beginning of this lecture, I said I'll give you a way to remember all the phases. Well, who do you pay your tuition to? Well, if you're a student at MATC, you pay it to MATC. And so if you remember this, I pay MATC, you'll never forget the stages of mitosis. I is interphase. P, I pay, P is prophase. M is metaphase. A is anaphase. T is telophase. C, cytokinesis. I pay MATC, and you'll never forget the stages of mitosis, okay? So if you always remember I pay MATC, you always will have the stages of mitosis in the correct order, okay? So there's your rule of thumb, all right? And so why do we need it? Well, obviously, I've brought the slide up before. We talked about it in replication and a number of other instances. Obviously, to grow to an adult, to fix any wounds that you might have, to fix any problems, you know, dead cells, things like that, and for plants to grow. Also, we look at mitosis as a way for cells to make new organisms. And so this is really beneficial, especially if you don't have a mate around. We unfortunately can't do that. We can't do asexual reproduction, but there are a number of organisms out there that do. And so what they do is they use the process of mitosis to make a brand new organism, break off of the parent, and form a whole new organism that way. And that's basically using mitosis as a way. You don't change the chromosome number. They're exactly clones, and we see the same thing over and over again. Okay, and the advantage of this, you don't need a mate in order to do these things. All right, so let's summarize. Mitosis is the process of cell division. We have the 4 stages: prophase, metaphase, anaphase, telophase. Prophase is basically when the cell gets ready, you get the condensing, you lose the nuclear membrane, and basically the centrioles move to the poles. Metaphase is when the chromosomes all line up. Anaphase is when they get pulled apart. And telophase is when the cell starts to look like itself again. You get the nuclear membrane reforming, the chromosomes line up, and finally you get this process called cytokinesis, which is where the cells divide. And again, the difference, animals, they divide with the cleavage furrow and pull apart, where plant cells form a cell wall or cell plate in between, and eventually that becomes the cell wall. And then finally, to answer the question, why do we undergo mitosis? Well, that's because we want to have cell repair, growth, and then some organisms will use it as a form of asexual reproduction. Okay? So with that, we've come to the end of the lecture. If you have any questions on any of these things, draw pictures, do these things, please ask. Stop me in lab if you want me to go over anything with you again and again. I'd be happy to do it and show you step by step what takes place. And for any other questions, please send me an email or anything else. And so I'll see you next time. Thanks.